Okay, welcome everybody. I think that we can start right now. I will make a welcome speech. I will give then the floor to the rest for the welcome remarks, and we will move to the scientific part of day one. Dear Mr. Livanos, Minister of Rural Development and Food in Greece. Dear Professor Zaphiris, Rector of the Cyprus University of Technology. Dear Associate Professor Karanikolas, Mayor of the Municipality of Naoussa. Dear friends and colleagues from academia and research units. Dear research associates and graduate and undergraduate students. Dear agronomists and peat stakeholders from the industry. Good evening or good morning, depending on where you are located right now. It is with great pleasure to welcome you virtually in the ICHS webinar series dealing with current trends and challenges on peach fruit production. Based on the initial planning, by this day, it was scheduled the inaugural meeting of the 10th ICHS Peach Symposium with physical presence to take place in Greece. Due to the COVID pandemic, we shifted the conference by 2022, 1 year from today, in the city of Naoussa. Naoussa is the epicenter of peach fruit production in Greece, with leading cooperative units with international impact and pivotal role in the canning industry. From a cultural perspective, Naoussa is a historical landmark since it is the place where the greatest philosopher Aristotle taught the greatness of classical Greek thought and the ideas of Platonic philosophy to the son of the king of Macedonia, Philip II, Alexander, and the other nobles of the Macedonian court. The webinar aims to bring together researchers from diverse fields of study who share a common interest on peach fruit production, covering the state of the art, spanning from breeding, development and testing of new cultivars and rootstocks, to fruit physiology and advancements on training systems, to pest and disease management. We aimed to bring all these disciplines together in a coherent manner. To this aim, I am glad hosting in this event plenary talks from top-tier scientists. The program includes 16 oral presentations distributed in 4 sessions, 1 every day. We do hope that the end result will be fruitful and productive, as it is reflected in the program of the current virtual symposium. The keynote presenters were selected to introduce and address the most important subjects in peach production, and I would like to thank all of them for accepting my invitation. Guido Snabel, Ksenija Gasic, Juan Carlos Melgar from the Clemson University of the United States, Thomas Graziel from the University of California at Davis, Ioannis Minas from the Colorado State University, Maria Fabiana Drinkovic from the University of Rosario in Argentina. Bo Zhang is representing the academia in China, the most important producing country of peaches all over the world. And now moving to Europe, our Spanish colleagues, Maria Jose Aranzana, Jordi Gine, Carla Casals, and Ignacy Iglesias. Our Italian peers, Laura Rosini, Davide Neri, and Guglielmo Costa. My colleague from France, Avignon, Benedict Kilot, and Nikos Papadopoulos from the University of Thessaly in Greece. We strongly believe that science and technology can further fuel agricultural and economic prosperity. The current virtual meeting aims at bringing together all recent advances and new knowledge regarding the cultivation of peach, which, as you all know, is one of the most competent cultivations. I would like to acknowledge the contribution of our commercial partners and the significant support that we have received, both by the local stakeholder as well as from global brand names. The event is organized by my institute, the Cyprus University of Technology, that is probably one of the newest state universities all over the world, located in the southern part of Europe, the cosmopolitan city of Limassol. The rector, Professor Zaphiris, during his welcome speech, will provide you a short overview of the advancements of our boutique university. I would like also to thank Mr. Christos Rodosthenos for coordinating and addressing all technical issues in a very professional and kind manner. I would also like to thank the ICHS for its critical input in this endeavor, and particularly Mr. Peter Vanderborght, Executive Director of the Society, for a really pleasant and smooth cooperation. Last but not least, I would like to extend my gratitude to the nearly 700 registered attendees for this webinar. We do hope to welcome several of them in Greece next year. Thank you very much. The next welcome talk is by the ICHS Executive Director, Mr. Peter Vanderborght. Peter has sent us a recorded message.
Dear Professor Manganaris, dear Rector of the Cyprus University of Technology, dear Minister of Agricultural Development and Food, dear moderators and panelists, dear attendees, I'm Peter Vanderborght, Executive Director of the International Society for Horticultural Science, and on behalf of the ISHS, I would wish to thank you for inviting me here on the occasion of the series of peach webinars that Professor Manganaris is hosting in his capacity of convener of the 10th International ISHS Peach Symposium that unfortunately, and due to the impact of the coronavirus pandemic, had to be postponed to May 2022. Having the peach webinars is a great and excellent opportunity to bring together a select panel of experts in a convenient virtual setting, allowing for maximum participation. These peach webinars will offer a great opportunity to keep the momentum going in anticipation of the in-person symposium next year, because let us be honest, even though we all embrace the alternative of virtual sessions, we must admit that each of us is looking forward with high anticipation to the moment where we can meet up, chat, and discuss with friends and colleagues again in person. I'm quite confident that the next 10th Peach Symposium next year in Naoussa, offering a high-quality scientific program in combination with an attractive social and cultural program, and an interesting 2-day post-symposium field trip will be a great success. For now, let us embrace the opportunity to participate in this webinar series, which I hope you all appreciate. I wish you all the best and look forward to welcome you all in person at the next year's Peach Symposium. Thank you.
I would like now to give the floor to the mayor of Naoussa, the city that will host the next ICHS Peach Conference next year, Associate Professor Nicolas Karanikolas.
Dear Professor Manganaris, dear Dean, dear Minister of Agriculture, dear friends and participants of the webinar. First of all, let me introduce myself. My name is Nicolas Karanikolas, and I am the mayor of the municipality of Naoussa. I also serve as an associate professor at Aristotle University of Thessaloniki here in Greece. Well, if we had normal conditions, I would probably welcome you all in Naoussa. Unfortunately, because of the pandemic, I'd just like to welcome you in Zoom meeting. Let's all hope that the end of the pandemic of COVID-19 is close to us, and that next year we will have the opportunity to meet to each other in Naoussa and discuss about our common interests, maybe with a glass of Naoussa's wine in our hands. I'm sure that Naoussa has many things to get from your knowledge and experience. I also truly believe that Naoussa has many things to offer to all the visitors and friends who visit us. Nowadays, about 33,000 citizens live in the municipality of Naoussa, and many of them are farmers or work at the primary sector of production. It's also interesting that our municipality exists from 2,000 meters altitude to a large area of plains near to 0 meters altitude. It's also well known that Naoussa has an interesting industrial past, as many forests, skiing center, and other hotspots for the visitors to enjoy. Fruits, gastronomy, the wines of the area are quite well known all over Greece and in some cases internationally. There is also a strong tradition and history for our town, and in Naoussa was where Aristotle, the greatest of the philosophers, taught for about 5 years Alexander the Great. But I'm sure that we can all discover all the beauties of Naoussa during your visit next year. I will be pleased to find you in Naoussa then. For this year, I hope that this webinar will be really fruitful and productive for you, and you will all enjoy your participation. As I discovered, you have a rich program covering many subjects of your scientific area. Thank you for the honor to give this welcome speech. Once again, I wish you all the best for your webinar. Thank you very much.
Thank you very much, Nikos. And I would like now to give the floor to the Rector of Cyprus University of Technology, Professor Zaphiris.
Thank you, Euro. Dear Minister of Agricultural Development and Food in Science in Greece, dear friends and delegates, it is with great pleasure that I welcome you to this digital conference organized under the auspices of the International Society for Horticultural Science. Looking at the program, I realized that it brings together in a multidisciplinary way the various different disciplines in this specific domain. Here at the Cyprus University of Technology, we strongly believe that science and technology can further fuel agricultural and economic prosperity. Our Department of Agricultural Sciences, Biotechnology, and Food Science looks at all these issues in a modern, integrative way. Our university is ranked among the top 350 universities in the world based on the Times Higher Education rankings, and is now a key partner in setting up the European University of Technology that will bring together 8 universities across Europe with substantial support from the European Commission. I congratulate the organizer, my colleague Yorgos Manganaris, and the team that assisted him in planning this event, and wish you all a fruitful and productive seminar. Thank you very much.
Thank you very much, Professor Zaphiris. And last, we have a recorded message from the Minister of Rural Development and Food in Greece, Mr. Livanos. And after this 5-minute welcome address from Mr. Livanos, we will then shift to the scientific part of the conference.
I would like to warmly thank the organizers and especially Professor Manganaris for the opportunity to open this webinar series on the current trends and challenges of peach fruit production. These informative peach webinars are held under the auspices of the International Society of Horticultural Science and represent a highly valuable online event. I am aware that this year's Peach Conference has attracted the interest of many people from here in Greece and other countries. Although it is really unfortunate we cannot meet in person this year due to the COVID pandemic, I would like to take this opportunity to invite you all to visit Greece next year for the 10th International Peach Conference, which will be held in Naoussa next June. Naoussa is not only one of the most picturesque and interesting cities in our country, naturally, historically, and culturally, but it is also renowned local in Greece, where the famous protected designation of origin Naoussa peaches is cultivated and produced. During the last year, we experienced the unprecedented COVID crisis worldwide. Our effort is to turn this crisis into an opportunity. Moreover, climate change is here. It is evident and absolutely urgent to get ready to confront its impact on environment, agriculture, and on everyday life. Last week in Brussels, during the EU Council meeting for the CAP Trilogue, I took the initiative to bring the climate change issue on the agenda. I presented the implications on farming, verticalization of production and exports due to the unusual incidence of frost last March and April in Greece, France, and Italy. This weather phenomenon has caused devastating effects on fruits, including Western Macedonian peaches, vegetables, and the wine sector, which, according to initial estimates, have suffered a huge damage. On top of my request to the European Union to provide immediate support to our farmers, I stressed out the urgent need to discuss on a European level viable, resilient, and future-proof mechanism to better protect and support our farmers and producers from events directly linked to climate change. These unexpected phenomena are projected to challenge the agricultural sector more often and more intensively in the near future. The primary sector has shown remarkable endurance during the pandemic, but there is also an absolute need for its holistic development through efficient and sustainable use of natural resources. In Greece, everyone has now realized the necessity to diversify the productivity model of our primary sector. The ministry is working hard in that direction, and we're developing our national strategy for the primary sector. Simultaneously, we plan to deliver our CAP strategic plan by the end of year 2021. In this context, I would like to share with you an initiative which is a central pillar of our strategy, the Greek Nutrition Project. The aim of the Greek Nutrition Initiative is to further enhance the production and consumption of agricultural products and food of higher quality. In addition, this goal is to highlight and protect their nutritional value and organoleptic characteristics of our agricultural products. Based on the indisputable nutritional wealth and benefits of Greek nutrition, this initiative will facilitate the adoption of good practices and promote innovation at all stages of the chain to implement the EU strategy from farm to fork. And peach is one of the most important Greek products, definitely the most important temperate fruit crop of our country. We're particularly proud of the premium quality of our products and also for our highly reputable canning industry. However, there are several challenges which need to be further addressed, mainly considerations for reducing production costs without compromising quality to make cultivation more profitable. In looking at the specific program of the webinar series, I realized that besides the scientific nature of the talks, several of the discussions are expected to offer valuable knowledge that can readily be implemented by the peach stakeholders. I have been informed by Dr. Manganaris that the webinar series will be attended by more than 600 attendees from all over the world, and I'm looking forward with great interest to receiving the key findings of this digital conference after the closure of your meetings. I would like to conclude by congratulating the organizers of this very interesting and productive event and wish every success to benefit from the knowledge sharing of following discussions. Thanks a lot.
Thank you very much all our esteemed speakers for their welcome remarks, and we are now moving to the scientific part of the conference. Each day we will have one moderator and 4 speakers. So at first I would like to welcome Professor Ted de Jong from the University of California at Davis. Ted is acting also as chair of the ISHS division of temperate fruit crops. He has 40 years of pomology research in California, focusing on peach tree physiology, peach rootstock physiology and production system, as well as work with other fruit and nut tree crops. Currently, he is working on a brief book on unifying concepts for understanding growth of fruit trees. Theodore, good morning in California.
Good morning, George.
So thank you very much for joining us. Our first speaker is Maria Jose Aranzana. I would like, first of all, to introduce her. Maria is researcher and team leader at the Center of Research for Agricultural Genomics, CRAG, that is located in Barcelona, Spain. After graduating in agronomy, she conducted a doctoral training in peat genetics and genomics at IRTA and postdoctoral training at the University of Southern California. Her area of research is in the field of rosaceae genetics and genomics, including peach, Japanese plum, apple, and pear. Her team is interested in different aspects of rosaceae genome organization, variability and evolution that help to elucidate the genetics of important agricultural traits. Their aim is to apply this information to develop tools as high-quality markers for assisted selection and genetic materials to improve plant breeding efficiency. Maria will present us today the impact of somatic mutation in peach breeding. Maria, thank you very much for accepting our invitation. The floor is yours for the next 25 minutes, and I would like to ask from our attendees that they have the possibility to raise questions through the Q&A session. It's the responsibility of Ted de Jong to select the most relevant of these questions and provide them to Maria during a 5-minute slot. If the questions are exceeding the time frame of 30 minutes in total, the rest questions will be answered through the Q&A session at the end of all 4 talks. Thank you very much, Maria. The floor is yours.
Thank you very much. I guess you can see my screen. Thank you. It's really a pleasure for me. Thank you for the invitation to talk about somatic mutations. I really think this is a very fascinating subject, and it's not only myself. Since many years ago, somatic mutations have fascinated horticulturists, geneticists, scientists. So I would like with this talk to transmit you a bit of this interest and this fascination. So we are all here because we all know that peach is a very important crop, and it's among the top 10 fruit trees. And every year, tens of varieties are released from breeding programs. Actually, to prepare this talk, I went to the CPVO database. I will see if I can use the lasers mouse. Okay, so I went to the CPVO database to see how many varieties were registered every year. So I could find data starting in 1995. So I found that in these 27 years, there had been more than 1,000 applications from 70 breeding programs. From these 1,000 applications, 75% had been granted, some are terminated, you can see here. But actually it means that in the last 27 years, more than 700 varieties have been in the market. These plots here show that, for example, that from 1995, 1921, every year there have been between 30, 70 applications. So the number of varieties of applications to get a registered variety have been very equal between years. And also I wanted to see what happened with other fruit crops. And I found very interesting that in apple, the number of applications was half in the same number of years, and even lower in other crops. So now I will explain very shortly a bit of peach domestication. So some years ago, we started a project to introduce in Spain some variability from land races from different origins. So I was in charge of looking at the variability, genetic variability. So you can see in these plots here that, can you see my laser? I don't know if you can see it. Okay.
Probably not, Maria. Probably
and
not.
I think it's freeze. Okay, fantastic. So you see that you see this pies. So there is one big pie in China, meaning that there was a lot of, the size of the pie represent the size of the sample set. And the colors represent the amount of variability, of genetic variability. So you can see, for example, that in China, there are many colors. China is the center of origin of peach. So lot of, here, see, I think I have a problem with that, with my pointer, but anyway, we'll see if we can follow it well. So this variability, you can see that the green variability is highly represented in all these Eastern countries, while some of other variability is different when we move along this side, go to the Occidental countries. And actually, the amount of variability, you can see that here, for example, the most represented variability is this brown here, is also represented here in the North American varieties. And actually, it's quite well in agreement with the distribution of the crop along the worldwide. Okay, it was also very interesting to see that here in the Brazilian varieties, we can see that there is this a lot of this yellow fraction, very similar to that in South Africa, and we could imagine that it was caused because of some commercial history between these two countries. So in summary here, we can say that the variability is different all around the world. Representing the variability, representing the accession that had been locally adapted to different regions. And actually, some years ago, in 1926, for example, I found this paper where they reported the use of Chinese varieties to solve some problems that they had with some disease. So the idea is that, you can read here, that there was a bad disease in peaches that were delaying blooming and foliation and were causing loss of crop. So they decided to cross them with some Chinese varieties because these Chinese varieties were resistant. So in the starting of these breeding programs, okay, the American breeding programs, they were introducing some Chinese varieties into their programs. And they generated offspring that was highly used in future breeding programs. So when we look at this variability, we see here that in the spice, most of the variability in the what we call founders, these varieties that were used intensively in breeding programs, have this blue variability. The commercial European varieties have also this fraction of variability, and also the American, meaning that in these breeding programs, we have lost a lot of variability. So if we want to increase variability in peach, we can even or cross varieties from different locations or use land races from other locations, or even go to cross with some species that are wild species of prunus or other prunus that are compatible and can be crossed. So regarding to this, my colleagues at CRAC, Perarus and Ivan Eduardo, are working in introducing, thanks to molecular markers, introgressing some genes, some traits into peach. So for example, crossing almond and peach, they have this hybrid that by selfing or by crossing with other peaches with several generations, they are obtaining a large amount of variability. So they could evaluate, for example, one of these progenies for 42 traits. I got from Ivan Eduardo these pictures showing this variability in the phenotypic variability of this the offspring. Also, these are samples when crossing peach with Prunus davidiana. But, sorry. I wanted to talk here today about somatic variability. Let me show if I yet can use this laser. Okay. So somatic mutations, when we talk about bud spores, we talk about lateral shoot, inflorescence, flowers that are different, that have a different phenotype to that of the original plant. So actually it's something that it's not new. We can find information of that from in Darwin book, for example, reporting that there were some plants or they could see some parts of the plant, branches, buds that show different phenotypes from the original plant. And actually this is caused by somatic mutations that can populate all the meristem or part of the meristem. So here I show you a picture of a meristem, a very bad draw of a meristem. I'm not very good at drawing, but in that at least you can see that the meristem is divided into cell layers, into 3 cell layers. The first is giving part to the epidermis, or if we talk in the fruit, to the skin. The second gives the mesophyll, or the flesh of the fruit, but also the germline. And the third is more the inner, the vascular system and the stone. So when we talk about a mutation in one meristem, we talk about there is a cell in one of these meristematic layers that has a mutation, and then this cell divides and populates the part of the meristem. But the way that it populates can change. It's not homogeneous, so it can, depending on the division. So it can, for example, there's here I show how it's only affecting one part of the meristem. So when this is growing, when the tree is growing, the branch is growing, only part of it will contain this mutation. Sorry. So you can see here that if in that case the mutation is only in layer 2, when we look at the leaf, only part of the leaf will contain this mutation. And this is really a nightmare when we try to do some analysis with DNA, because when we extract the DNA, we are not, and we want to compare the DNA of the mutant and of the original one. At the end, we have a chimera. We have a mixture of genotypes. And actually, what it is. It's a chimera, that it's a heterogenomic organism that it's composed by cells of more than one genotype. However, if this mutation occurs in layer 2, then it will be transmitted to the progeny. But if not, if it's only like here, only in layer 1, these fruits, the seed of this fruit, are not going to transmit the mutation to future generations. However, we are talking about peaches, trees, so we can just cut and propagate. Okay, so this means that somatic mutations are really relevant, especially important in crops where we can have clonal propagation, and in trees because they stay in the field for many years and can accumulate more mutations. Here, I show you one way of division of population of the meristem, but could be others. So in that case, it was only affecting one part of layer 1, but could affect all layer 1, or could be sectorial, affecting the 3 of them. So here, I show you a model of what can happen if several layers are suffering mutation. And this is something reported by Cheng in 2015, where the original one is a white flower. The mutant, if there is layer 1 mutated, then we have a pink flower. And if we have layer 1 and 2, it's a red or darker pink. And we can have also these chimeric, these variegations, depending on which fraction of the meristematic layers are changing. Actually, we may think that probably this is something low frequent, but no, this is highly frequent. It happens. Sometimes, I mean, it's necessary to observe very well the trees, but it's occurring. So here we have, it happens often in grape, but also in citrus. And I show you here some pictures that, for example, this is an underdeveloped peach with part of the skin being nectarine and the other part being a peach. Also, this is a peach where the original was white and the mutant, it's a chimeric on the yellow color. And this here, I think it's very funny because it's a a commercial variety, Silver King, a mutation from Arm King, that it was yellow. And we see that the suture keeps being yellow, and suture comes from layer 1, while layer 2 is white, meaning that in this case, the meristematic layer that changed was layer 2. This chimerism was explained with some surprise some years ago in 1924. It was very nice for me to find these papers. It was funny. And in that case, this was a mutation, a sport that was ripened 3 weeks later. What happened here is that if you observe this black line here, is not really a black line in the suture, is red, is ripened. So the suture ripened at the same time than the rest of the peaches in the orchard, while the mutation in the layer 2 didn't allow, or was the one producing this delay in ripening. So Weldon wrote this paper reporting some observations from some growers, some curiosities, but a couple of years later, Chamel and Pomeroy did a more exhaustive analysis in that case by analyzing more than 7,000, observing and deeply phenotyping more than 7,000 peach trees from 11 varieties. And they found up to 39 mutations, some of them in combination. So it was 39 mutants, but some of the mutants had more than one differential trait. So some were early, some were late, but then accumulated some changes in Princeton, Clingstone, or other traits. Actually, also these authors a few years later reported a big frequent of mutations of sport mutants in fruit trees. So they reported 146 in peach, but more than 1,600 in other fruit trees. And curiously, 32% of them, or of the plant patents, were implying these sports. And in that paper, they also talk about the need to be careful because sometimes some of these mutations were undesired. And by accident, a bad branch, could be replicated and propagating orchards with undesired traits. And actually, it's fun because they report this, or they say that this large number of sports, patented sports, could be motivated by the fact that at some point there was one variety starting, well, one sport starting from delicious, that rise a high price because of that mutation. So all growers started to look very careful their orchards. Also, recently, in the book of Oki, of Handbook of Peaches and Nectarines, I went through this book trying to spot the varieties that had been annotated as mutations. And I counted more than 170, so for sure I missed some of them. Meaning that actually before, in the early years, and now spore mutants have a high impact in breeding, in the use on commercial peaches. But not only in breeding, also for research. Because you can imagine that we have two trees, two individuals that are identical except on few traits. So this awakes of interest from scientists. And for example, some of the mutants that have been broadly studied are those nectarines coming from peaches. Because usually these nectarines have some other phenotypes that are considered pleiotropic effects. So they are rounder, smaller, they contain more sugars. So there were two hypotheses. One was that with this mutation, the mutation changed more than one gene. And the other hypothesis was that probably the gene that had mutated intervene in many other biological processes, and at the end was changing other traits. And actually, this is a possibility because the gene producing this nectarine, the loss of pubescence, is a transcription factor, so it may be involved in other processes. Also, it seems that some mutants from peach, for example, Yuval, that it's a mutant from mother, that's a peach, has different response to cold storage. So there have been studies of the transcriptomic studies of this behavior. Or, for example, in this paper, they look at some traits comparing one peach with 3 of their mutants. In that case, they also confirmed that the peaches were bigger, the nectarines were smaller, but also more sweet, or with more sugars at least. But the 3 nectarines were a bit different. So they hypothesize that probably the mutation are a bit different between them. And actually, this was published in 2016, but a few years earlier, we had sequenced with Illumina some of these mutants. So here, for example, you can see Odet and Yuval and some of these flam prints nectarines. And actually, we already observed different levels of somatic variability. So these are using some filters, and we thought we look at how many polymorphisms we found between pairs of peach and nectarine. However, at this point, what we did is we sequenced leaf. And as I said before, leaf is a chimera. So actually, knowing exactly the level of variability here, it's a bit tricky because we need to play and understand and find the right filters to be able to know exactly the amount of variability. Another mutation, or that kind of mutation, it's from flat to round. And it's something that has been reported back in the early 20th century. And we found that mutation also, we started to study this mutation also in 2012, 13. And it was a flat variety that changed to a round variety. And actually it came from a tree, and one branch showed this novel phenotype. So we decided we wanted to study at the genome level what happened. So we ran some markers that were associated with the flat trait. So we knew that when we found the 464 allele, so this peak here, it was flat. And actually, what we found when we look at the leaf DNA is that faint peak here. So we didn't know if it was a contamination. And then we thought about the possibility of having a chimeric mutation. And actually, when we extract DNA from the skin, that it's layer 1, we had this flat, the same genotype. And we only observe change when we look at the flesh. And the stone also share, the DNA from the stone also share the flat, meaning that in that case, mutation happened in layer 2. Then we looked at the whole genome level, and we sequence, as we realized here that when we use the leaf, we only found a small contamination of the other layer. What we decided to do is sequence whole genome from leaves, and we compare the variability in the 2 genomes. So here you can see that in that, this is chromosome 6, where this gene, the flat gene is located. The flat gene should be around here. And we observe that the level of variability, this is number of heterozygotes, loci or SNPs or fragments in this region, is similar between the flat and the mutant. But when we come here, the mutant has lost this heterozygosity. It's almost homozygote. So we can say that this part here, that we see these faint peaks, are because of the contamination of layer 1. And the hypothesis here is that in the original one, we have one of the chromosome, the father chromosome, the mother chromosome. And in the mutant, this mother chromosome broke, and was replaced with the original, with the other one, with the father, meaning that in this region we only have homozygotes. So just to finish, I want to give you these take-home messages that somatic mutations have awakened the interest of horticulturists, growers, breeders, and scientists, that mutations can be advantageous but also undesirable. And in those nurseries that are in charge of multiply some varieties, they should take care of the branches to be sure that it's stable. Also, that chimeras are very, very frequent and need to be taken into account, into consideration when we do genomic studies. And we find some papers where it's not the case. And only those mutations in layer 2 will pass to the progeny. So if the mutation is in layer 1, the only way to transmit it, to transfer to the offspring, is by cutting and grafting. So I want to thank I want to thank your attention, of course, and also to my colleagues at the Genetics and Genomics group in the Rosasi. And part of this work was done by Elena Lopez many years ago. Now she is researcher at the Plant and Food Research Institute in New Zealand, and to Sang Yu, that is postdoctoral in China. So I'm open to questions, and thank you very much.
Thank you very much, Maria. Very interesting talk. Appreciate it. I have a couple questions that people. One question was, are there established populations between segregating populations between nectarine and peach?
Excuse me, if they are?
If there are established, say, segregating populations between nectarine and peach.
What do you mean segregating population? I mean, if there are, I don't understand. Sorry, I mean, there are sport mutants that are commercialized, if this is a question, and they are stable as the mutant. The mutation is stable in some cases, of course.
Yeah, and breeders or geneticists have been studying crosses between nectarines and peaches for ages, right?
Yeah, yeah, I mean, these are, there is only one gene of difference between peaches and nectarines.
Right.
So there is a single gene responsible for the loss of the pubescence. So this works like a regular genetics. I mean, you cross a peach and nectarine and you are crossing actually two varieties.
Okay. Another question, you mentioned about the peach nectarine chimera. How do they inherit? I guess, how are they inherited? And do they stay stable and consistent through the season? Or after a few generations of propagation vegetatively, do they change?
Yeah, in some of the papers that I show you from the old years, they did this progeny test, like was just cutting and keeping. Usually they are. So of course, if they are chimera, it will depend on the part of the tree, on the part of the branch that has been affected, and on the tissue. If it's just by cutting, you have to, it's, I mean, usually they are stable, so you just can cut and graft it. And if the buds that you have selected have the whole layer mutated, then you will be able to propagate it. And actually, as I said, I mean, there are many varieties that come from these somatic mutations. If they are chimeras, then it will depend on the part of the branch. And I have seen some sports, some mutants, that even if it was, for example, from flat to round, depending on the part of the branch, the round was more, it was not that round, so it was more oblate. So meaning that when we are talking about chimeras, we need to observe, I mean, to observe very carefully what's going on, and there is not a rule.
Okay, we have another question here. Do you have any somatic mutations of peach where it just went to fuzzless from regular fuzzy peach to so in other words, basically have nectarines been derived from mutation of peach?
Well, I mean,
The somatic mutation of peach.
yeah, well, it's when a mutation occurs in layer 2, this mutation it's transmitted to the offspring, but obviously mutations can occur also in the germlines directly. So we are talking about different things. One thing is a somatic mutation. So if nectarines, what we know, come from a somatic mutant, we don't know. It could come from a mutation that occur in the germline directly due to meiosis, right? So when we talk about somatic mutants, usually we can find some traits that are observed in some varieties or some individuals that arose from variation, some mutations in the germlines directly. In that case, for example, in the case of the flat peach that I show you, it's not because it was a new mutation that was introduced to change from flat to round. It's because a part of the chromosome was replaced. So then one gene or one allele was removed.
Okay. I have one last question before we go to the next speaker. Are there environmental or horticultural conditions that are known to induce bud mutations?
Yes, I mean, well, it's not, we know that in some conditions, it's more, for example, when there are this in vitro culture, cayuse culture, there are mutations, somatic mutations. So when the plant is subjected to some stress, we may think that there are, could induce some mutations. Also, when the plant is subjected to some other biotic, abiotic conditions, stresses, it could happen. The way that if it's studied, how it happens, how it occurs in field, I'm not sure. One thing that for me it's very interesting, but it's just a feeling, is that some varieties have more tendency to produce mutants. So I'm not sure if this is true or only some varieties have been observed or only some breeders have observed. But my feeling is that there are some varieties that have this predisposition to have some changes. So if this is caused from some epigenetic reasons or some transposable elements that are changing from some mechanism of DNA reparation that are a bit different, we don't know, or at least I don't know.
Yeah, very interesting. It certainly seems like it, because sometimes we have whole lines of varieties that came out of one original. Yeah,
well, actually, the chimera that I showed you, that it was a peach, that it was white flesh and yellow flesh, is the same variety that came from flat to round too.
Oh yeah. Okay.
Okay. So.
Right. Thank you very much. Very
Thank
interesting.
you very much, Maria. Thank you. Just to let you know, the rest attendees, we have some questions that can be answered by Maria at the final sessions, the Q&A session. So Maria, we have some questions. You can check them on the list and you can give a response at the end of all talks.
Thank you very much.
Thank you very much. The next speaker is Ksenija Gasic. So I would like kindly to ask Ksenija to share the screen. And to make
George, you need to allow me to start my video if you want to see me. And
you're allowed already, Ksenija.
Okay.
You can share. You can.
Now it allowed me. Thank you.
Okay.
Let me just a second.
Okay. In the meantime, I will make a short presentation of Ksenija. Ksenija Gasic, she is professor of horticulture and a peach breeder and geneticist at Clemson University, located in South Carolina, the United States. Her program is developing fresh market types of peach varieties adapted to environmental conditions and replant tolerant rootstocks. The emphasis of the fresh market breeding is on combining high quality and consistent productivity with improved resistance to diseases such as bacterial spot and brown rot and enhanced health promoting benefits. Rootstock breeding is focusing on combining tolerance to two replant diseases that affect the southeast of the United States, namely Armillaria root rot and the peach tree short life. Dr. Gasic's research involves characterization and utilization of peach genetic diversity and development to improve breeding efficiency. Ksenija today will provide us an overview of the advances in peach cultivar and rootstock breeding. Ksenija, the floor is yours.
Thank you. I believe you can see my screen. Thank you, George, for the kind invitation to present here and to organize this webinar, which is a really nice way of segueing into the next year meeting in person. So as George said, I will try today to cover the advances in genetics with a specific overview of what's happening in peach. And to do that, I'll try to cover the resources that are available, potential that they bring, and we'll focus on the breeding for disease tolerance in both cultivars and rootstocks, and we'll cover 2 diseases for each. So cultivars, bacterial spot and brown rot, and for rootstock, peach tree short life and Armillaria root rot. I will try to close with some potential future perspectives of that these advances bring into the breeding. So we know peach belongs to Prunus genus and Rosaceae family, and the genus Prunus includes many horticulturally valuable species. But peach leads the group with the genetic resources available, and thanks to the resources developed for peach, many other crops within Prunus and Rosaceae have been making some big advances. And in this slide, you can see that almost every single crop within the Prunus genus has its genome sequence, except the tetraploid tart cherry, which actually helps with some downstream applications that I'll touch upon briefly in the next slides. So peach, why is the peach such a good example or a reference for our genome or genome for the Prunus crops? Is because it has a relatively short juvenile cycle, it's self-compatible, which sometimes can be a problem. It's diploid, it has a relatively small genome and high phenotypic diversity. Although, as Maria Jose just presented, the genetic variability is not that high, which forces us to go into the relatives either within the Prunus genus or even outside of it to try to increase the variability. So what's very important for the peach, Prunus genus, and the whole Rosaceae family is that there is a high synteny and compatibility within them, which allows us to use knowledge gained in one to apply into another, or even to make interspecific crosses to gain some additional knowledge. The resources for Prunus and Rosaceae are held in the genome databases for Rosaceae, and there are many markers, linkage maps, genome sequences, QTLs, and so on. But the major achievement of our research community was sequencing the peach genome. And because the way we use the homozygous Lovell diploidized genome, we actually have an extremely high quality genome sequence that has been used for many discoveries since it was released in 2012, and later on, as this citation shows, in 2017 with some improvements. In the meantime, Prunus community also worked hard on developing some general platforms for genotyping to provide some unifying scene for comparing research data from different groups and different locations. So we released the basic first 9,000 SNP pitch array in 2012, and then recently improved it with additional 8,000 SNPs, which makes our genotyping work much more uniform across the groups. So many of these things are already summarized in an excellent paper led by Maria Jose, where we actually went into detail about the effect of the genome sequence availability on the prunus genetics and application. But before that summary, there were two major projects in prunus or on peach or in rosace species. One is Pruvidomics in European Union, and the other one is Rosebreed led by United States, that develop many of these tools that we are using today, and some of which I will be showcasing later in the talk. And I'll just showcase one as an example of many new advances that are coming almost daily and being published in the scientific journals, as with this one, which showcase the comparison of over 300 genome sequences to look into the specific changes in the specific regions, and particularly of the horticulturally important traits. So I like to show this map that Elizabeth Dirlewanger published in 2004. And at the time, those were the QTLs available for Prunus genus. And you can see here by coloration and coding that majority of them were mapped in peach. So you can imagine 16, 17 years later, that picture is much different. And yesterday I searched the GDR to see how many QTLs are reported overall in Prunus, and there's almost 1,700 QTLs, and about 1,000 of those were reported in peach with 83 unique traits. So you can imagine what resource we have to actually go deeper and provide some tools for breeders. So if we show the picture of just one piece of the peach genome, chromosome 4, you can see how many more of the traits, and I did not even put all of those that were mapped, are mapped on top of each other within the same region. Similar stories in other prunus, like in this case, I'm showing the sweet cherry, where it has the same region in the chromosome 4 and the overlap of almost the same fruit quality traits. What that allows us is, because we have genome sequence for peach and for sweet cherry, we can compare the regions of interest from two species and see if there is a difference or similarity, or maybe for some traits that are mapped in one of those, or one of the species, in this case peach, we were showcasing all of the traits mapped here in the peach, in the cherry genome, to see is there a variability or difference between them. So one of the problems for the breeding programs is the timeline. So from the moment we make a cross to the moment we release the cultivar takes about 15 to 20 years. The idea of these advances being applied into the breeding is to help breeders choose the parents, to know about the traits that parents possess, and predict the combination of traits that you can expect in the progeny. That the DNA information can help us choose the combinations of these parents to create a specific outcome. And of course, the final piece of the puzzle is which seedlings to progress. And we would want to have the tools that would allow us to do that as early as possible at the stage of seedlings in the greenhouse. We don't have to wait 3-4 years to see the feature in the field. So all of these are geared toward increasing breeding efficiency. Basically, we all know that the breeding is a numbers game. Probably many of you heard that line many times. With these DNA tests that research and Prunus and Peach community is developing, we are trying to increase the number of seedlings that we create, because we can decrease the number of trees that we have to evaluate in the field if we apply the DNA test in between and basically cull the seedlings that we don't want and plant only those that have a potential to have a trait of interest in the field. So now I'm going to go down into the detailed stories about the cultivar and rootstock advances, focusing on the disease resistance or tolerance. So one of the diseases that are problematic here in the southeastern United States, where my peach breeding program is the bacterial spot. The problem with bacterial spot is that there is nothing growers can do if the year is favorable for the disease. There are some mitigating approaches they can do in the spraying, but nothing really would prevent susceptible variety developing the symptoms. And you can see how devastating it can be to a crop and also to the whole tree, because it affects the leaf and the fruit. The disease affects both. So through the Rosebeet project, we worked on mapping the bacterial spot tolerance in both leaf and fruit. And then because of the fruit being more important for growers to being able actually to sell the fruit, we focused first on developing the DNA test for the fruit tolerance assessment. And in this slide, you can see how we were able to develop the test that is very cheap, less than $1 per sample, including the DNA extraction. It can be done in-house on the regular endpoint PCR machines. This is a CASP-based test. So basically, we are detecting the SNPs that are distinguishing between the different alleles susceptible or resistant. And we have just 2 test runs that we need to run to basically discriminate between the susceptible and resistant. We are using this test fully in my program. And what I've actually been doing, I went backwards and I tested all of the material that I moved into the higher levels of selection to see how well I was selecting for disease tolerance, along with actually the fruit quality and all of the other phenotypic traits that I was evaluating in the field. And basically out of the about 5,000 seedlings from the 2016 to 2020, we selected 37% of them had the potential to have a tolerance of a bacterial spot. Last year, fortunately or unfortunately, we actually had the outbreak of bacterial spot at our farm. And I was able to confirm in the field that this test was almost 99% accurate, which makes my life much easier, because now I can actually focus in the field on the trees that are potentially having the tolerance to bacterial spot. Another disease, which is not unique to southeastern United States, but the whole world, is brown rot. And when we tried to map the brown rot and looked into our families segregation that you can see on these 2 slides, average for the families was not that great, but we found that there were some individuals that are showing really high tolerance, even for skin or for the flesh. And here you can see how they look, because the tolerance to the disease was in the Bolinha, which is the landrace from Brazil, which has a horrible horticultural quality, but has the tolerance to brown rot. So we had to do some introgression and trying to increase the quality in these selections is not that easy, because it requires back crossing with the high quality parents. So when doing the mapping, we found out that many small genes across the whole genome with a small effect were associated with the tolerance to brown rot, and which led us to try to explore the genomic prediction to see how we can use all genome markers to predict the behavior of the individuals for brown rot tolerance. When we apply the different models in the genomic prediction, we use several different families that have Bolinha as a tolerance source, and the contender also, which was found out with the Pacheco et al. Paper published several years ago, that contender also carries some level of tolerance to brown rot. And we used each of these families separately to predict for others, and in combination, we found that there are several different models that would behave better than the others. Then for those particular models on the right-hand panel, you can see we applied a different type of cross-validation called leave one family out, to just see if they would still show the high predictive ability. And basically, overall, for both non-wounding and wounding, non-wounding referring to the skin and wounding referring to the flesh, disease severity index, BSA and RKHS models showed superior or the same predictive accuracy than all of the other models that we used. So now we're basically using the genomic prediction to advise me when I'm doing the cross-planning. And what we were pretty amazed and pleased about is that majority of our phenotyping was done in lab, because that's much easier to control and manage. And we were worried that what we data used in lab might not be predicting correctly what's actually happening in the field. So we phenotyped the individuals that were phenotyped them both in lab and in the field. And in this slide, you can see how well the lab data is predicting the field brown rot tolerance. And you can see that using just the lab data, we were able to distinguish between the material that has the potential to have a low or high field disease incidence. Basically, high disease incidence meaning a low tolerance. So this paper is currently in review, and we are hoping to actually extend that to some other traits that were polygenic and with lots of genes with a small effect that we want to apply in the breeding. So now moving for the last section of the talk to the rootstock advances. In the rootstock, as George already said in the introduction, we have two issues in the Southeast. One is the peach tree short life, which is a complex syndrome that includes sensitivity to the ringed nematodes, and then a scion of the rootstock and scion to the bacterial canker, as you can read here in these bullet points. And we managed to solve that problem by developing the guardian rootstock, which tolerates the nematodes, and the trees survive on the soil. But since we did that, and majority of the orchards were planted on guardian, we realized that we actually have a second level of problem, which is Armillaria root rot fungus that grows slowly and takes 5 to 7 years to actually kill the tree. So to develop the resistant rootstock from the southeast, we need to have both. We need to have the peach tree short life tolerance and Armillaria tolerance. So on the side of the peach tree short life, we sequenced the guardian genome and compared it to the actual reference genome, which is of the Lovell, and Lovell was used for decades as a rootstock in peach, to compare the regions, the QTLs that we found on chromosome 1, 4, and 6, to see if there are some differences. And what was interesting about this is that previous mapping for tolerance to ring nematodes localized the region on the top of the chromosome 2, but you can see here that we did not find that. We found it on the chromosome 1, 4, and 6, which actually corresponds with the regions that we detected when we mapped the bacterial spot for fruit and leaf. And that directed us to think that this actually shows the response of the scion to the Pseudomonas. And we are now exploring this and trying to develop the DNA test that would allow us to do the selection based on the peach tree short life. And then, of course, the Armillaria. Armillaria is a really devastating fungus. You can see here how the orchard on the replant site looks. There's almost nothing alive. And you can detect if the culprit is Armillaria if you scrape the root and you see the white mycelia. Here you can see the progression through the time of the same land, how many and many trees are vanishing, and most of it due to Armillaria as well. So to address that problem, we were successful on getting first this project funded, and now currently last year funded the second project to try to find the solutions for this Armillaria problem, because very little was known about the problem, and it was really hard to assess the tolerance of the material because there was no way to do the quick studies. So we developed this resistant pipeline when we actually evaluate the tolerance in vitro. Then we check the we use root microscopy to check if the fungus is penetrating the roots. And on these slides, the red is the root structure and the green is the fungus. As you can see, these are the tolerant responses when the fungus is basically lining out on the outside of the root and not penetrating. And then we developed the co-cultivation system in which we grow together clonally propagated material on the different, inoculated with different fungi, and using the foam structure to mimic the structure of the root similar to the field. We use this system to actually investigate the transcriptome changes in the challenged roots by fungus. And we compared susceptible and tolerant rootstock and found that there was an induced and constitutive response, and all of the tolerance coming from the plants. Now, we found a few candidate genes, and right now are working on engineering Armillaria resistance in Guardian, because basically what growers would want is to get the Guardian with Armillaria resistance, so they don't need to change how they grow the trees. I'm at the end of my talk. With the future perspectives, you can imagine with the thousands QTLs reported in peach, those are endless. And because of the Rosaceae family and Prunus genus are so synthetic and collinear, that provides us an opportunity to translate the horticultural and disease trait information across species into modern breeding pipelines. Thank you very much for your attention. And I won't have the acknowledgement slide, as I acknowledge people that work in the particular sections on each slide. And there are too many people to thank, specifically with the Clemson University Peach team and Muscle Fruit Research Farm that where the breeding is happening, and all of the members of my lab that are helping with this. So that's all I had, and I'm open to any questions.
Thank you, Ksenija. So if anybody has any questions, quickly type them into the question and answer thing on the screen. I had one. You were looking at brown rot resistance with Bolinia. I mean, in the background, have you found resistance in anything related to anything but the epidermis or the skin of the fruit? Because Bolinia is this awful thing with lots of fuzz and.
Yes. So what I found out is that we will not have a resistance for brown rot. I'm thinking more like in a line of having high tolerance and helping growers modify their spray program to not need to spray the whole season, but maybe just a week or two before the harvest. That's going to be the story with the brown rot, because we do have individuals that show high skin tolerance if the skin is not damaged, because that provides the opening for fungus. And the second has a good flesh tolerance, but not good enough to not allow fungus to initiate the damage, if I'm saying it correctly. That's not save growers. That's part of the problem, because if just the small damages on the fruit, you cannot market it.
Right. Okay. That's why I'm curious, because I know Tom has worked on that for a long time and so on. So one more question from Julio Pisano. Are you using the crosses of some of the cultivars of North Carolina, such as Norman, Candor?
Excellent question. I did not, 25 minutes was not enough to mention in details, but yes, the brown bacterial spot resistance comes from, oh my God, my brain is now, from North Carolina cultivar that you didn't list here, and my brain just stopped here. Oh God. I'll remember and I'll type to you. And because of the rose breed and because of the material that we genotyped with the SNP array, we were able to look into the haplotype diversity in the whole US breeding material to see if there are other haplotypes, not just those that we found in the cross that we made, not contended. Oh, God. Between the North Carolina cultivar that's resistant and the O. Henry. We use that material as a segregating progeny. And we found that there are two resistant haplotypes or alleles, and both of them are in the North Carolina material. And the same one, allele 1 is more predominant, and allele 2 is, let's say, specific for Arkansas breeding program. And they draw their material mostly from the New Jersey breeding program when they establish and start. So that's how the mapping of the allele across the material in the United States showcased itself. But what's important to know is that there are two regions, chromosome 1 and chromosome 6, that are involved with the fruit response for bacterial spot. And chromosome 6 region is dominant and more important than the chromosome 1. So even if you only have a resistant allele on chromosome 6, you achieve the good field tolerance of the material against the bacterial spot.
Ksenija, thank you very much. We
Thank
have
you.
several questions in the Q&A session, so you can take your time to check
will
them
do.
out and provide your response at the Q&A session at the end of all talks. So thank you very much, Ksenija, and I would like to give the floor to Davide Neri. Davide is a professor of arboriculture at the Department of Agricultural, Food and Environmental Science at the Polytechnic University of March in Ancona, Italy. He received his PhD degree in tree physiology from the University of Bologna, Italy, and his main area of research is fruit tree propagation and nursery technology, pruning and draining, flower differentiation, pomology and germplant evaluation, agroecology, root physiology, sustainable orchard management, and the replant problems related to soil sickness. Since 2015, he is consultant for biodiversity and fruit production at FAO, being the focal point for the Italian report on biodiversity. He has also served as director of the Italian Fruit Research Center. Davide will provide us today his lifetime experience to describe current trends and challenges in training systems of peach. Davide, the floor is yours. You can just unmute. Okay.
Okay, just I want to stop here. Okay, ready? So, current trends in training system. What is the main problem now? Is to face several threats against the peach cultivation. So we need to face the climate change, and the systems should take in account that we have to make our orchards more as a net sink for greenhouse gas emissions, and this means that we have to change the way to manage the soil and improve the quantity of carbon that we are able to store in the soil. And this is a big threat for the type of cultivation. So we need to make a sort of better intensification systems that are able also to face this problem. Then we have to work with the reduction of the use of the labor. So we have to work more with mechanized system, maintaining the quality of what we are producing. So this is a very important problem for Europe, and we can consider that now the Green Deal for the next 10 years will face, the sustainability as a main problem for the production of the food, so including the fruit species. And this must be done with the minimum impact and the possibility to work with mimicking nature as much as we can. So we are asked to reduce in the next 10 years the pesticide by 50% and the loss by nutrient by our soil of 20%. And finally, we have to increase organic farming up to 25%. So this is a very difficult task to be reached in such a short times. For sure, we don't have problem with antimicrobial. So when we speak about the systems for training the peach, now we have to work taking into account soil fertility, landscape, and finally the peach orchard as a way to produce fruits. This means that we have to work with a better comprehension of the role of soil organic matter and to better agricultural practice for carbon sequestration. Then we have to work with the soil fertility, increasing the capacity of the root system to get the nutrients from the soil. So it's quite complicated. And this means that we have to better understand root growth, first of all, and the physiology of the tree, mainly with this relationship between shoots and roots in working together in the interaction. And this means also that we have to work with root system that is able to work with byproduct amendments that will be our duty for the next years to be included in our cultivation system. Then compost quality and possibly some type of mulching. And I will propose a solution using living mulch. Then we have to work, in two different main systems that are organic, so ecological, biological systems, or integrated system, sometimes called also conventional agricultural systems. And this must be done in a view of circular economy, and moreover, including the role of biodiversity that we have in the landscape, in the soil, and in our cultivation systems. So this empty soil should be a problem for the future in this view. So we have to work with complex systems, and we have to find a way to make the intensification of our cultivation systems more sustainable. So we have to work in some way, including some ecological engineering of our soils and our cultivation systems. And this will be, in a very narrow situation that should maintain yields while reducing external inputs. So we have to improve our ecological replacement, or we have to enhance yield without enhancing external inputs. That is a not ecological enhancement. So these very, very narrow windows in which we are able to work is the sustainable intensification that we have to go on in the next future. So here we can see this, progress from the 3D orchards to the 2D orchards that was presented by Iglesias Ignacy, that will explain, I think, the next talk in the next days. And this seems something natural, something very easy to be done, but in this very deep change from the systems that are more natural up to these very intensive systems, we have the problem that root system for the peaches will face the problem to have some allelopathic interaction that will be negative with the same genotypes, and so without diversity. So we have to include in our system the possibility to make the root system working well and improving the possibility to make its evolution and absorption and capacity to explore empty niche in our soils. And this means that we have to improve also our way to use these amendments, organic amendments, and to make the root system able to get these results and to get these nutrient sources without making a failure of their behavior. As it was done in this case, it was good results with olives. So this means that we have to work with a system that is able to humify the organic residues with different sources of organic matter, with different microorganisms, so to make also some way to control and to limit the spread of some soil diseases that are very dangerous, as we have seen before. So we have to learn how to manage better the soil fertility and the capacity to use organic residues, getting some good results as it was done here with green compost, and including the great diversity in the soil and great use by organic microorganism in the soil. So here you can see that we have some models that can help us. So we have to escape from monoculture. We have to go to crop rotation, but with our peach orchard will be very difficult to make crop rotation. So maybe we have to work here somewhere with mixed crop system that can help in going towards a more natural soil condition. That was done in other species. This is the grape pine in my region that is cultivated now with experimental orchards that becoming more and more popular, with more than 6 hectares now planted with the wild strawberry as a cover along the row. So sort of living mulch that is able to reduce the competition with some very aggressive weeds and reduce the capacity to have soil erosion or something like this, because we protect the soil and we cover the soil. So that can be done also with peach, and we can see some example in organic farming using a very high diversity by the natural cover that can be managed along the year and can create the possibility to have the root system that is working in a higher diversity and on the same time a good production. And here some green manures that can be also used. Here are some new orchards that are just planted and they can grow with different clovers or mint that can be used in the soil. So thanks to biodiversity, we can go on with training system. That means that we have to work with a system that have something that is moving from a geometrical to functional shape with minimal pruning. And this must be done with some possibility to work with equilibrium with root system and the aerial part, avoiding the senescence, maintaining the plant in this reproductive phase with an equilibrium between shoot and roots that is devoted to production with some efficiency. So we can describe the growth of our orchards, working with the possibility to make this vegetative phase and then reproductive phase. And so we have to think that if we use dwarf rootstocks, we have to reduce this vegetative phase. And so this will mean that we have to do more for the root physiology, because on the other hand, it will be just a big problem to make to maintain this in the future. With vigorous rootstock, on the other hand, we have the problem of economic loss that is derived by the delay in reproductive phase. So we can go through this early production with some high density orchard, and we have to work with rapidly with the reproductive phase. But this means that we have to manage this with some recurrent pruning, and this pruning can be done in a different period of the year, not only in the winter. And it will be very, very, very useful to be done in at least 3 windows along the winter. And we have to avoid any mistake to increase with the winter pruning the vigor too much, and maybe then after we will not be able to control this anymore, and so we will have a loss of production. Then we have to work with the pruning system that is must be more efficient than precise. It will be easy way to manage the pruning with some very mechanically intervention that can create the possibility to reduce the amount of labor for a unit of peach produced. So which kind of solution we have? We can work with traditional base shape or with central axis, and this will different way to get these two results. And we can work more with using the physiology of the epinasty, and this will create the possibility to make you know, a self-organization of the of the tree, and this is possible to be done in this way, working with the control by the upper part on the bottom one. So this is something that was used in different training system to make this open base or this central axis very efficient, and that they use some head cutting that are very popular now to open the base and to open the branch according to the training system in an easy way, and sometimes also with a long pruning, so without with alternating the head cut and the tie long. So that is possible to control the opening of the shoot without making a lot of effort in the pruning system, and this can create the possibility to manage the branch in a very efficient way. This is a curvilinear branch that can work properly, and this is a linear inclined branch that has trouble in our condition to create more water sprout and less fruiting shoots. So this kind of solution is much more equilibrated and give more fruit per unit of pruning material. In this case, we see the possibility to manage in an easy way the formation of the central axis system. You can see how we present this to the farmer and how they can manage this in different cuts and organization. So to get the final goal, to have a tree that is with a central axis well organized in a conic shape and very efficient in giving the maximum production in the bottom part. And this is something that can be very efficient in managing the production. This is the possibility to have also a very nice control of the light distribution inside the tree, and giving some type of pruning that is like mouse tail pruning, and that can be easy done by any pruner, any worker that can create this in a very efficient way. Also, according to the possibility to use the regularity that we have inside the pitch, and this is due to the pilotax system that is 2/5 on the pitch stem. So working not with the head back of the main stem, but working with the natural way to manage the shoot by the plant itself, we can have a really efficient way to manage this central axis. If the vigor is too high, is higher, we can use, or medium vigor, we can use a B-axis system that is quite common now, or if the vigor is higher, we can even use a candelabrum with 3 axes. So, or the traditional palmette. So to make this wall system, this edge system with single central axis, we can work to increase the production in the early years using tie long, so a long pruning without head back of the laterals. But this can create trouble to the lateral shoots that can have poor quality. So for this reason, we have to increase the way to distribute the light inside the system that are also protected by the presence of these hail nets that will be more and more common. So to control these, we need summer pruning, at least 1 or even twice, using the possibility to mechanize this system and make, in this case, cut and the new growth of the shoots that are equilibrated and well lit. So this create efficient way to make a wall with the possibility to introduce also the capacity to reduce the spray of the pesticides because the hedgerow is very thin. So in this case, you can use several type of different pruning cuts and in relation to the quality of the shoots and the variety. So in this case, it's also very important to avoid any strong lateral branch in the top part. And so this must be cut to maintain the situation under control. And this is also the possibility to have some more free situation, which is a small central leader that can be very efficient in creating a very high production that can be managed by the ground or with just a platform that is only with one layer. In this case, pruning must be done to have some control of the self-shading. We used several years the Y shape, but generally speaking, was changed in open small Spanish vase. That is something that is possibly mechanized to with summer pruning to make the new laterals forming very quickly. But with the final pruning at the end of the season, in September, we can prepare well the plant before the blooming that will be in early February or March, and this will make possible to have, a control that is done completely by the ground. This is the pruning small Spanish open vase, and with the possibility to have a much lower labor intensity per ton of material. We can work with 3 different periods of pruning, so winter to create vigor, and during the summer to control the light inside the plant, and this will create a very efficient system, but not expensive, because we are able to control also mechanically the growth of the shoots. Then we can have the control also the quality, because we can decide which kind of shoot to maintain, and in the position we will work with the fruit set and fruit thinning. That is something that is very important. Obviously, we have a big problem with climate change, and that will be something that needs to have some active action that we have to do to control our systems. And one is the use of hail nets. And this hail nets is something that was done also with different color. So different photoselective nets are very common now in our fields, in our orchards, because the quality of the light change according to the color, and we can have some different results in the field, and different quality and different control of the and different shading. And this is possible that for the farmer to decide which kind of solutions they can have in their orchards. And according to this, we have also the possibility to control the UV radiation. So we can think that we have a reduction under the normal hail nets that is about 20%. Moreover, we can create some orchards that are waterproof to avoid the cracking of the fruit or some disease like gray mold, or we can have the control against insects that can be controlled. So what we expect for the future, just go on with agile systems. They are easy mechanized. They are going to control and to be easy covered with the photoselective nets. And we can work using a better soil fertility management. This was a conclusion that was done already in 2013, and there was repeated more or less in 2017, giving more effort on the photoselective nets. But I think that now is very important that the next on 2017 will be more and more on soil fertility management to improve biodiversity in these intensive training systems. Thank you.
All right, thank you, David. We don't have much time for question. I'll just, I have one here. It's talking about, they're concerned about leaf mulching and the occurrence of dangerous insects to create airborne phytoplasma.
Okay, that it can be a problem. Obviously, it can be a problem in some climate, in some condition, and you have to change the management of these leaves and mulching according to the real condition of your environment. So I suggested these two species in my environment, but the project that we did in Europe, it's obvious. We have to find in each possible solution the right species to make the cover. So we have the duty to cover the soil more. That is what they asking to agriculture. We have to avoid organic matter mineralization, and we have to cover the soil. We have to make the root function better. So for doing this, we have to try and to research in each situation the proper soil cover.
Okay, thank you very much. I have one real quick question. I think they're asking if it's necessary that the hedgerows be oriented north and south.
Yes, yes. In our climatic condition, is very important to address the rows in north-south, because we need to have the better light distribution inside the canopy as we can. So the better the light distribution, the better the production, and the easier way to manage the orchard. So I would say try to do your best to make north-south.
All right, thank you very much. I think we need to go on to the next session.
Thank you very much, Davide. If the attendees have any question about you, they can write it down in the Q&A session. And moving to the last talk for today by Dr. Ioannis Minas. Ioannis is an assistant professor of pomology with the Department of Horticulture and Landscape Architecture at Colorado State University. He has received his PhD in pomology and post-harvest physiology from Aristotle University of Thessaloniki in Greece. His research program focuses on the characterization of the influence of genetic, environmental, and dorsal factors on fruit tree physiology, resilience, and productivity, with emphasis in pitch. And his research integrates techniques from core fruit tree physiology, cold hardiness, fruit quality, non-destructive sensors, post-harvest technology, ripening physiology, and systems biology. Dr. Minas currently serves as the coordinator of pitch rootstock trials within the frame of the USDA's multi-state project North Central 140. Ioannis, the floor is yours.
Thank you, George. Thank you for the invitation, for a great organization, and really looking forward to come back next year in my homeland. Actually, my homeland is like 15 minutes from Naoussa. So I'm looking forward to also a very promising event in Naoussa. So I'm going to be talking about the true impact of pre-harvest factors on peach fruit quality, development, and metabolism today. So some general introductory information. Peach per capita use in the United States is in a constant decline over the last 40 years. Consumption is failing constantly, and we have recorded in 2015 in a pretty low rate of consumption, 2.8 kilograms per capita in the United States. So this trend further drives reduction in peach production. Let me put my laser pointer here. So further drives this trend of reduction in fruit production. Poor consumer quality, also some labor issues in mainly California are causing that trend. And this is observed mainly in fresh peaches and fresh nectarines, as you can see with the red and yellow color over here. So a little bit about the status in the world. China is by far the major producer, followed by Spain and Italy. And then my two halves, Greece and United States, are competing actually for the fourth position. In the States, California is by far the major state in production, followed by South Carolina and Georgia. My state, Colorado, is ranked 7th in terms of total production, but is when you estimate the value of the production per the different industry within the different states, California again is by far top, but you see here Colorado is, instead of 7th, it's been ranking 5th in terms of value of production. So why this is happening? This is happening because our growers are able to receive premium farm gate prices. And you can see in that graph, Colorado is by far top in farm gate price per ton of produced peaches, close to $2,200 per ton, and California, the top producer, is ranked last. So to summarize that, national farm gate average price is $1.2 dollars per kilogram, and Colorado growers are receiving $2.2 per kilogram. That $1 difference is giving them an extra 15 million that I see that as a quality premium, because our peaches here in Colorado, because of the growing environment, high altitude, can achieve high levels of sugars and quality, but also the focus of the industry is more to the local markets and neighboring states are targeting high harvest quality and optimal maturity in order to maintain that quality premium that they receive. So we have two things now, two messages that I want to pass. There is a trend. Consumers don't like the peaches that we deliver them, and they are not happy with them. That consumption is going down, but there is also a big amount of consumers that are willing to pay the extra dollars for high quality fruit. So in a previous work that my mentor Carlos Chrysostom with his colleague did earlier, asking consumers why they're dissatisfied with peaches, they received a mix of responses. To my eyes, this kind of responses is like consumers don't like peaches at all, at least whatever we offer them. And even some of the responses are related with insufficient post-harvest management. We have to keep in mind optimum post-harvest management is only here to make sure that we are not going to detriment the quality that we develop in the orchard. So if we want to improve fruit quality and peach quality, the only way to improve that is at the orchard level. And to emphasize that, I'm just using a slide that I'm using in my class. And this model of how quality is built up in the orchard. So we have the source sink system, sources of photosynthesis products, and sinks, which is a fruit, shoots, and roots. In that case, because we're interested for fruit production, we're going to focus only on the fruit. But of course, The other parts are playing role in that optimum quality development. So in the sources, in the leaves, light and CO2 together with water are being captured and through the calving cycle are being transformed to sugars, mainly in peats, sorbitol and sucrose. And these sugars are being translocated to the sinks to be further transferred to monosaccharides, and these further catabolized through glycolysis to develop volatiles, pigments, proteins, metabolites, other kinds of carbohydrates, acids, and I have a question mark with starch because peat is not storing too much starch, but if you want to use this as a model for pommes as well. So all these carbohydrates are constituting what we call dry matter content, which I'm going to talk later about it. Previously, we have reviewed what work has been done in terms of pre-harvest factors that are affecting peach fruit quality. And that figure, it was what we used in that review paper, and is giving you the whole overview of how many parameters can affect peach fruit quality at the orchard level. In my talk, I'm going to be focusing in crop load, which I find the most important parameter to affect peach fruit quality, and fruit canopy, fruit position in the canopy, and rootstocks. So let's see how fruit quality, how peach fruit quality is changing during peach fruit development and ripening on the tree. Here we have a bicolor cultivar, which is June Gold. This is from a previous work that we did in Greece. And here in the bicolor cultivar, you can easily see the change of the green color to the yellow and orange color in the background color, not the red blast that you see, but just underneath of that. And during maturation, that change in the background color is reflected in that graph with the ground skin color, with a hue angle that is declining together with firmness. So fruit, as it's maturing, it's softening. And then CO2 is increasing, ethylene is increasing, and drives further that softening process of the fruit on the tree and ripening. Acidity is going down, dry matter content and soluble solids are always increasing while the fruit is attached on the tree. So that gives us another message that the more you leave the fruit on the tree, the more sugars you get on the tree. There is no invented boxes that can provide sugars to the fruit after you harvest it. So the more you leave the fruit, of course, then you have to make a decision how long you're going to be transporting the fruit in order to harvest them at a certain firmness to make it to the final destination. Another message that I'm going to highlight here is that fruit quality is also changing and improving with the advancement of maturation. And a lot of the studies that I saw in the previous slide haven't really take maturation into account when they were studying the impact of pre-harvest factors on fruit quality. So fruit quality and maturity assessment requires specific tools and also trained people to do that. The standard for the industry for the determination of maturity is flash firmness, and that is also an index of shipment and storage potential. Soluble solids concentration is, or BRICS, is giving a sweetness index to the fruit. And then dry matter content, which is a fairly new index that we are using in fruit systems. It hasn't been studied too much in peaches, but in other fruit species has been correlated with sweetness and consumer acceptance. Just one more highlight, these tools are, these methodologies are destructive and labor intensive, and also they are not very easy to be used in the orchard for immediate measurements and decision making. So for that reason, we have been focusing on the development of non-destructive technologies to estimate internal fruit quality in my group. So we're using 2 handheld non-destructive sensors to estimate internal fruit quality and maturity in the field. And we use that for a larger fruit volumes that will help us to understand the effect of pre-harvest factors on real time. So we're using the F750 produce quality meter, which is a near-infrared spectroscoper, an open type instrument that allows for on-site calibration. And we have been very successful with dry matter and soluble solids concentration at 729 to 935 nanometers. This device allows for 3 online measurements at the same time, but only 2 are displayed. The second device here on the right, the orange one, is a product of the research of Dr. Costa within Bologna University and his colleagues, is what we call DA meter, which is also a visual near-infrared spectroscoper and is a closed type instrument. That means that it's coming calibrated in the factory. This device is giving you the index of absorbance difference at the levels where chlorophyll absorbs the light, and it's kind of an estimate of chlorophyll content, kind of an estimate of background color, and it's really, even in the bicolor cultivars, we can see the background color. That is not the case with the fully red cultivars, and over there, this device can be very handy. And we have seen over time that this parameter is related heavily with fruit physiological maturity. So how we measure fruit quality non-destructively? We use near-infrared radiation that enters the fruit, the light scatters and interacts with the fruit tissue. Then the light absorbance is measured by the spectrometer within a few seconds, and through a data processing step, the many raw absorbance values are converted to a single prediction value. The advantages of that approach is the fruit is not distracted. We can use it in the field. We can have a faster measurement, increased number of samples to increase our understanding and help our decision making. And we can also optimize the models that we're using. The disadvantage for most of the industry members is that the models must be built to use the instrument. There is no commercial accuracy sensor right now in the market. To build a model, you have to make sure you will use a broad variability for your calibration samples, and that hasn't been done in the most studies that have been published. So we developed recently and we published a calibration protocol. As you see here, we develop canopy crop load levels in order to increase the variability of internal quality, but also we selected fruit for our calibration population across 5 developmental stages. So this way we were able to have maximum variability, but also variation within the same developmental stage or similar states of maturity. We also scan the fruit at the different developmental stages at different temperatures to include storage, retail, and field conditions, respectively. So doing independent validation analysis, we saw that through our novel calibration protocol, we were able to accurately calibrate near-infrared spectroscopy for dry matter content with a pretty low error, 0.4, which is like a third or a fourth of what has been reported so far. And then soluble solids concentration, 0.6 error, 0.5 to 0.6. And then we were also able to incorporate readings of the DA meter in this open source device. So we are hypothesizing a novel concept device that can assess pitch quality and maturity during fruit growth development and at harvest in the field and during post-harvest as well. So with a single scan, you can take these 3 measurements with that device that we are trying to develop. One other interesting finding during our work with calibrating near-infrared spectroscopy is that we have seen that different varieties, because of their different textural properties, are being segregated based on their spectral profiles, and that is done in a manner of in an order of their ripening time. You can see here These are the spectral profiles of 7 cultivars, and there is a segregation of them across the time of ripening. Even though there is textural properties that are affecting spectral characteristics, our calibration methodology was really strong and has a lot of a lot of variability in the models that allows us to calibrate many pitch cultivars and even optimize them for dry matter and DE meter. So we can continue our work in the field. So using the device, we went back to assess the effect of crop load on Sierra-rich pitch dry matter content at harvest. And as you can see here in the distribution of values, dry matter is provided as in cluster categories. The unthinned fruit, you have a peak at 11 to 12% dry matter. Most of the fruit are presented in that category. And then the more you thin, you have a shift of the population towards higher dry matter levels. At the commercial stage, you have increased further, and then you have at the light, the more even you thin, the more quality you obtain from the fruit. I also here highlight, I'm presenting dry matter, but soluble solids with dry matter in that variety, and mainly in pitch, they have very high correlation. Similarly, we did a study to see the impact of crop load on pitch fruit maturation at harvest using DA meter. DA meter, when the fruit is immature, it has higher values, and as maturation process goes, the value of the index of absorbance difference is declining. So in the unthinned fruit, you have a peak of more than 1.2 in DA meter value. Sorry. And then the more you thin, the fruit are maturing. And with the light, you have a peak at 0.2 to 0.39. So that also demonstrates how dramatically crop load is affecting quality, but also maturation. And when we do comparative studies, it's better to make sure that we control for maturity. And this is what we did. I will just put a note over here that DA meter, maybe someone will ask later, does not directly correlate with firmness if you plot individual values, but if you cluster the values with different categories of index of absorbance difference, you get a high correlation. But we have found DA meter values describe better physiological maturity on pitches than firmness. So we use DA meter to control for equal maturity, or our concept device that is at the same time is measuring index of absorbance difference, in order to study in depth two distinct thinning severities, two distinct crop loads, and study the quality, but also the non-targeted metabolomic profile. We use a concept of carbon sufficiency, commercial thinned pitches, and unthinned, which was fruit that were starving from carbon. At this developmental stage, maturity was equal between the thinning treatments. So as was expected, fruit from the thinned category, from the carbon sufficient, revealed superior quality at harvest when compared to the unthinned treatment. And that was also highlighted in a PCA analysis demonstrated over here. Fruit quality parameters were segregating better fruit samples at the later stages of maturation. So you see here at the early stage at S2, there is no big difference in terms of phenotype between treatments, but they are very different. The quality and the phenotype is very different at the end of maturation, at harvest, at S4. Surprisingly, we saw that when the fruit were having the lowest phenotypic differences, so they had the largest differences in the metabolome. You can see here at S2, we saw the biggest distinction in terms of metabolic profile between thinning treatments. And when they were similar, when they were distinct in terms of phenotype later in advancement and maturation, the metabolome was pretty similar. So many metabolites associated with the primary metabolism shift according to development process. And we saw minimal differences because we are also selecting fruit at equal maturity. But at harvest, we saw only 2 compounds that they demonstrated significant differences. This was between the thinning treatments or the carbon starvation treatments. In the catechin was highly present in the thinned treatment, and an unknown compound that is related with stretch, it was correlated. It was found in high levels in the unthinned treatment. So we correlated catechin by selecting the different samples at harvest. We correlated positively the catechin to quality parameters, which is dry matter content and soluble solids concentration. In contrast, the unknown compound was correlated negatively with quality. So the hypothesis for future studies from this work is that central metabolism is not changing too much when you control for maturity. It's really genetically programmed, but the fruit, when they are in sufficient carbon supply, early in development, they can invest in luxury compounds like secondary metabolites like catechin that can be utilized later on as a source of carbon in order to optimize, to have an optimum pitch phenotype. And it's a kind of a priming effect early on in order to develop a better fruit quality. So we use the sensor in order to see what is the impact of fruit canopy position on internal pitch quality at harvest. We selected 2 cultivars. The one that I'm showing over here, it's a Sierra Rich, an early and very weak cultivar. And we selected 3 canopy zones where we scanned the whole lot of fruit in its zone with our sensor, measuring at the same time DA meter and dry matter content. As you can see, we control our results for equal maturity based on DA meter. And we found no differences between canopy zones when we control for maturity, because we hypothesized that a low vigor cultivar allows for enough light in the bottom of the canopy in order to have an optimum quality development. We did exactly the same in a more vigorous cultivar that was not allowing too much light in the bottom of the canopy. And then we also control for maturity. So all the zones data were controlled for the same DA meter value. And we saw optimum quality was achieved only on the top of these 3 canopies. So we saw significant difference across positions because high vigor situation is causing non-uniform light across the canopy. We did mesocarp and exocarp metabolism, GCMS metabolism analysis, and we see similar variation across position for each cultivar in the mesocarp. We hypothesize that mesocarp is affected by environment, is less affected by the environment because of exocarp might be protecting, and also changes in the mesocarp are heavily regulated by development. When we study exocarp metabolism, we saw what we were expecting. Vast differences only in the high vigor cultivar, where we saw the less uniform light condition. We try to model all the changes that we find from this study in the mesocarp and exocarp on fruit positioned in the top or in the bottom, or more light or less light. And in summary, we found sorbitol, sucrose, and catechin indicative of superior quality, metabolites of superior quality. Amino acids, citric acids, and monosaccharides like sorbose and fructose are indicative of inferior quality. The last case that we tested was the effect of rootstock on peach fruit internal quality. These are data that are being that were taken from the NCN40 trial that Greg Riegert was coordinating. was planted in 2009, and in 3 seasons, 2016 through 2018, we studied that with equal maturity control and also crop load control across the different tree vigor situation. As we know, the larger the tree canopy, the more fruit you get. And this is what we found, even I'm not presenting this data here, but we saw a significant effect of a trend on dry matter content. The smaller the canopy, the more dry matter you have. As you can see over here, with red is the trunk circumference area and dry matter. The larger the tree, the more vigorous the rootstock, the less dry matter you get. We further did the GCMS analysis, and we found that rootstock vigor and light availability are highly correlated with the internal quality. And you can see here the less vigorous rootstock. You can see the arrow here is showing the change from dwarfing to vigorous rootstock. The less vigorous rootstock had higher content of dry matter, soluble solids, but also higher levels of available light. And that was also reflected in primary metabolism. So primary metabolites were shifting across the different vigor situations. So in conclusion, near-infrared accurately assessed dry matter content, soluble solids concentration differences in pits at 729 to 935 nanometers. DE meter does not correlate with firmness, but describes better fruit physiological maturity status. Adaptation from the growers is really behind because it requires calibration, high cost, research and development departments. This concept device that we have already filed a provisional patent and we're considering moving ahead with patent can help for simultaneous maturity quality assessment. We can use this for research to better understand the true impact of pre-harvest factors, but also comparing fruit at equal maturity. Crop load severely affects internal quality, maturity, and maturity at harvest. Early stage fruit metabolite responses to distinct carbon supply prime peach fruit quality. So early thinning, as Ted was explaining with his previous approach, we confirmed that even with metabolomics, so we couldn't compete him. Excessive tree vigor, as it is related with cultivar or rootstock, and fruit position in the canopy affects light availability and fruit quality. So use of summer pruning and or rootstocks, semi-dwarfing rootstocks, when they will be highly available in reliability to facilitate uniform canopies for optimized light availability and fruit quality. So I would like to thank my lab members and collaborators for this work, and also the funding agencies and collaborators from the industry and stakeholders that are making sure that we're going to make peach great again, starting from the United States. Thank you.
All right, thank you, Ioannis. So you used all your time, I think. Yep. So maybe we have about a half hour for questions from anybody and everybody. I'll just start with Ioannis because we just finished him. In the meantime, anybody who wants to ask questions of any of the speakers this morning, I think you can. The person in control of IT will allow you to speak if you raise your hand. So if you want to speak and ask a question, raise your hand. I have several very nice talk, Ioannis. I have several questions for you. I have one specifically, the distribution, fruit distribution, fruit quality distribution that you showed in response to your crop load follows very similarly to the fruit size distribution that we saw with crop load. And I'm just wondering, have you ever tried to correlate just size distribution with your quality characteristics?
There is a correlation, and crop load will affect size. and will affect internal quality. But in the rest of the studies that we did, we were using fruit of equal size and maturity as well. So I didn't mention the size, but yes, crop load will affect size. This is the main thing that we.
Yeah, well, we know that for a long time. I was just wondering if it.
We have the data just for the heck of the time. Yeah, there is the same trend. There is the same trend. There is always that distribution, canonical distribution, normal distribution across, and in the extremes you have a different distribution. But in the middle,
curiously,
size is similar.
We found that even for when you thin down to like 50 fruits per tree, that distribution does not go away.
But we saw another interesting thing when we thin. That's a really good point, Ted. When we think crazy, like the light, 1 fruit per shoot, we saw increase in size, but quality, internal quality, dry matter and sugars are not decreasing.
Oh, that's very interesting. Yeah. Okay, anybody, let's see. I'm not sure whether the moderators are opening people up for questions, but I have a question here. Can you harvest the peaches any later after the DA meter measurements hit near 0 values?
I think when they hit 0 values is like close to ready to eat ripening. So for the different cultivars, that's the one problem with DA meter. For the different cultivars, you have different optimum levels. So in different regions, research must be done in order to adapt DA meter for the different values. As you saw in my slides, one variety we were harvesting for same firmness levels at 0.7, and the other variety, a later one, was harvested at 1 DA meter for same firmness levels. Later varieties we have seen, they tend to have higher DA meter values for optimum harvest.
Yeah, okay, thanks, good. Does peach skin thickness affect the NIR analysis?
Could be that the reason that we don't see good correlation in some cultivars. In some cultivars, we don't see that. So I haven't really studied the skin thickness, but we know that near infrared cannot go very deep in the fruit.
All right. Do you have any correlation between ethylene concentration and NIR values?
There is demonstrated by Costa et al. Ethylene is correlated with DA meter values when they are clustered as well.
Okay.
We have also a question from Professor Bassi. How much are dry matter and soluble solids related?
Highly in peach, 99%.
Okay.
That's why I like dry matter more. Now we're setting up consumer trials, but that's why I like, because these kind of devices that they have already in the market, this prototype from Felix, which actually you take it without any model, you just have to calibrate your models. It's working better for dry matter. So it's actually measuring water content, and we estimate the opposite of water content.
Okay.
Well, we have one from Lorenzo Leon. Is it better to develop independent NIR calibrations for each cultivar?
So that is what has been published and has been reported. What we see, we are getting really good. We have developed 3 models, major models, one for bicolor early, bicolor late, and full red color variety. And we selected based on the characteristics of each cultivar, which model we're going to be using for this variety. We saw, even with just selecting models from previously developed models from different cultivars, they were highly accurate, but then we were able to optimize those models for different cultivars. So there is, I will not say the main atmosphere is like you have to calibrate for every cultivar, but with our protocol development, we don't need that. We just need to optimize per the different cultivars with 100 more specimens.
Okay, yeah, thanks. So we have So Rodrigo Infante saying he's seen high variability in DA meters values within the same fruit measured on both cheeks.
That is one problem of the device setting. So I don't know how better we do that when we use a larger lens. I'm not like, I agree with that, there is variability. And also light is affecting, like ambient light is also affecting the reading. So if you have to be careful where you measure that. So if you're pointing against a light source, that may affect your readings.
All right, thanks. We're going to go back to the beginning.
Thank you. Thanks, Ted.
Ted, just to ask from Christos to spotlight all speakers and eliminate myself. So Ted, you will have all speakers and you can start the questions.
Okay, thanks.
Thank you.
So I have a question from Jose Chaparro for Maria. How do you differentiate between homozygosity and hemizygous in the case of Pinto to round sport?
Yeah, initially when we talked that if we find difference in the heterozygosity in that branch of the chromosome, it could be because of a deletion of the chromosomes. In that case, we talk about this hemizygosity. And we thought about this option, but the depth of sequencing was the same. We didn't find a reduction on the depth of the sequencing. So that's why we thought that actually we were having this. And also we were able to find the breaking point too. And we could amplify the region where the chromosome broke. So it helped us actually to verify, to validate that it was really a chromosome repair with the other branch, with the sister chromosome. Hope it's clear, Jose.
Yeah. Okay. Let's see. Oh, I do have one. Is there a possibility to stimulate artificially somatic mutations to create new cultivars? Well, of course, they used to do a mutagenic exposure to radiation and all sorts of stuff in the old days, but.
Yeah, I mean, this is the known option. If it's more direct thing to do, right? Or with in vitro cultivation, but not in that case. So it just would be the mutation, the chemicals and radiation. Yeah. I think there was a question also if there is any case of reverse mutation from nectarine to peach. Yeah. I haven't seen it.
Okay.
So it's not, I haven't read anything about that. So no.
I'm sure Ksenija can tell you why that is. I was curious with David. You didn't mention dwarfing or size controlling rootstocks and the role they're going to have in the future.
I didn't mention because I am really afraid for peach about the use of dwarfing rootstocks. So I think that the future is for medium vigor, equilibrate rootstocks, but not very low vigor. So I think that the problems in managing the tree in the field will be much higher than the advantages. So I would suggest to use more the other techniques to manage the growth. Like also Ioannis noted, the summer pruning is very efficient in controlling size and quality and the uniformity of the results in the field. So if we are able to create these systems that are easy to be managed with some mechanical summer pruning as the 2D system, so the narrow system, I think that we are able to get the control of the vigor enough to create an improvement in in the use of labor and the quality of the fruits. And also we will be able to have the possibility to cover with the hail nets and also the waterproof hail nets. So that mixed system that is increasing now for the also for cherry. You cover the central part of the edge, and then you have on the lateral you have the hail net. So you cover with a waterproof plastic, and then you have the laterals. So I don't need exactly a dwarfing rootstock for peach in our condition.
Okay, well, our growers are very interested in size controlling rootstocks because they need to control vigor. We have really vigorous growing conditions, good soils, but I agree with you. You can't go, if you go too far, you're in trouble. Like in our case, controller 5 is way too dwarfing, but some of the intermediate ones that we have, I think are pretty good. I have a question for Ksenija. Could resistance to bacteriospot be associated with the number and size of stomata? Thanks for an excellent organization. Oh. I think your speaker's off. No. George.
Yeah, but probably we do not hear Ksenija. Ksenija, can you please try?
Yeah, she just did.
Probably we have a problem with Ksenija. No, we cannot hear her. So
Ksenija, can you check your microphone when it's connected to the computer?
So probably we have a problem with the microphone of Ksenija. So apologies, Maximiliano. We have to move to other question. Or
now
Ksenija,
better.
Now you can hear you. Okay.
I mean, it was the same. I just had to uncheck it and check it again for some reason. Computers are not my friends today. Anyway, we did not look into the number of stomata in relationship to the bacterial spot. And if I remember correctly, Dr. Ritchie from North Carolina was our bacterial spot guy when we started looking into, and he is the guy for the bacterial spot disease. He did not mention it, but might be. I actually had a question for Ioannis, but because we are panelists, we cannot type into the question box. So there were several questions that floating in my mind, especially if you tried slow softening cultivars. Because relating to the question about DA meter hitting the 0, because those slow softening, they are 0, but they are firm and you can keep them longer on the tree to increase the quality with the sugar accumulation and everything. And DA is gonna sit at the flat 0.
Yeah, good point on that. We haven't done any slow softeners. In Colorado, we use only traditional cultivars because they are more hardy. That is the setting of the industry. But it will be interesting. We still think for the industry, we have done through our work, we have done discussion with many stakeholders. There is a resistance to adopt DA meter. It's not easy because it requires a lot of other information to finalize your decision. And still they feel firmness is the optimum. So we are reevaluating our approach for firmness. So far, near-infrared hasn't been successful to be analyzed through near-infrared, but we are taking different avenues.
Well, that's interesting if I can respond, because several years ago when the DA meter hit the market and I started evaluating it, we found out that, of course, you have to stick with the yellow flash versus white flash because the values are completely different and the readings you get. So the standard white, yellow flash cultivars, not slow softening, not a regular softening type, that's majority grown here in the Southeast, they have a 0.6 to 0.8 value when they're perfect for harvest and will sustain the packaging and everything. And so one of our biggest growers is using DA meter to manage his big orchards and to plan what to harvest and when.
Yeah, I agree. This is something like locally also has been done. Maybe the light environment in the different growing conditions will affect DA meter performance also. Like our values here, we are 1,500 elevation, so it will be different from what you see over there. But so far for these standard peach cultivars, even full red color, DA meter is consistent, and we feel very safe with that. But these kind of other versions, yes, I agree.
Ioannis, one more question for you from Professor Grappadelli. Any merit to including fruit vascular flows in fruit quality studies?
That's a good point. And we have been trying to do something with the dwarfing rootstocks and vigorous rootstocks, but we are still early. I have to squeeze my students more. No, I haven't done anything on that, but it's something that we're discussing.
Okay, so we have also one question, go back to Davide, about orientation of orchard. Is the geometry of the orchard that makes impossible the north-south orientation, or it is better to move a vase training system, or it is possible with cultural techniques to have a profitable 2D orchard even with a west-east orientation? Can you please open your microphone?
Yes, thank you. Thank you, George. Yes, you have to discuss about the geometry and of your orchard. I think if you can't make the north-south in our type of climate, I think it can be better to use a small open base that can be very efficiently done also in the other orientation. So it is much easier. I would say that with peach, it will be quite complicated to maintain the production in the bottom part with the wrong orientation of the rows.
One follow-up, can I ask you, Davide?
Yes.
So you mentioned from the question of Ted about dwarfing, but what about 2D systems? What is your overall perspective? Even utilizing moderate vigor by diffusing the vigor by more scaffold. So what is your opinion about 2D in peach, 2D systems?
I think that is a big opportunity. So it's a big opportunity if you have the condition to make an equilibrated tree with the distance between the rows between 4 and 5 meters, and the tall of the tree that can be 3, 4, 4.5 meters. So in this condition, you may get some very good results, and I think is a very important opportunity to be used in relation to reducing the use of chemicals and the use of fertilizer, because in this condition you have an easy way to control the the growth and the mechanization. So finally, it's important to make this also for the climatic control. So what is actual situation is that we have the duty to make a protection against hail, against the possibility that you have sunburn in some situation. So you need to have the cover of the orchards. So in some way, at least in our condition, I would say that we have to go to high quality fruits in these 2D systems. Obviously, when I work in a different environment, maybe hillsides or some more protected areas, I would suggest to use the small open base. Is very efficient in making the labor organization and the fruit quality. But I would say that if you have a farm that is in intensive area like ours, with with our social condition, obviously, it's better to have the 2D system.
Okay. So I'm wondering if we left any question unanswered, especially for Maria Jose. From Jose Chaparro, I suppose all of them have been responded, right, Maria? Okay, excellent. So I'm wondering if anybody else would like to set some questions. Otherwise, I will ask from Ted to make some concluding remarks and close day one of the webinar.
Anybody have any other questions like George asked? Hearing none, I guess I'd have one comment before I make some remarks on what David is saying. I know in California, we have gone through the going to vertical systems, not really 2D the way they are done now, but those type of systems, and all our growers have pretty much are going back away from those because of the excess cost of establishing the orchard. And we tend to have more corporate farms in California, so they're really very interested in bottom line. I'm not saying the way we do it is the right way, because most of our growers are going broke, but I think the small open vase or a minimally pruned open vase is more the thing where our growers, we have not a huge amount, but many of them are trying a system that Kevin Day has been promoting, where we try to maintain a tree no higher than 2.5 meters, plant them a little closer, and going with a modestly size controlling rootstock.
I see.
Yeah.
We have some more questions, so Ted, give me a few minutes more before your concluding remarks. We have from Dr. Arous a question for Ksenija. Ksenija, do you think that the genomic selection approach you use for monilinear resistance can work well for other quantitative characters like sugars or fruit weight?
It's an excellent question. We are exploring that because the number of markers that are needed is not that high. It's about 500 what you need in the given material that you have. And since peach is so closely related, we have no issues with the reference material to predict the material in the breeding families. And so we are really trying to explore that possibility. I don't have the data to show you. I know we did some test runs with the rose breed, and we showed that it can be fairly highly predicted. But in my program, we focused on the bacterial brown rot for the genomic prediction right now.
I do remember there was one question that disappeared, but there was a question about resistance to Tophrina deformans, peach leaf curl.
Oh yeah, I answered the typing. We do, okay, so in South Carolina, Tophrina is not a problem. So although I come from the region of the world when it's a bigger problem. And there is a material in a former Yugoslavia landrace, we call it vineyard peach. There is like maybe 5 or 10 accessions that have a complete resistance to Tophrina. And we actually have them in the repository in California. when I got a position in Clemson, I moved them, and you know how long it takes to go through quarantine, but they're finally there and available for work. And I got that question often, and it should be fairly straightforward. And I know Thierry Pascal from INRA in France and Benedict, they worked on the Tafrina, and I don't know where that research is, close to publishing, I believe, but they have some interesting findings. So it's going to be much easier than brown rot.
We have also a question from a graduate student from my department in Cyprus. Very interesting question about low chilling varieties. In Cyprus, we need low chilling units, about 200 chilling units. So can we advise for a particular type of tree shape, rootstock? Can we correlate them with the fact that Cyprus, like Sicily, are characterized by low chilling units? Davide, can you have an idea about that?
That is a nice, important problem, because generally speaking, they have a low chilling and also generally an upright growth, very vigorous. So it's really important to follow this orchard according to repeated green pruning. It's not summer pruning, because you have to start earlier than summer, you know, because it's still spring, which you have to work with different green pruning. And I think that make possible the solution to have the small open base easier than the vertical system, the 2D system. But in any case, whatever solution you take, I think you have to follow with repeated summer pruning. And obviously, the integrated system with water management and the possibility to have the nitrogen applications that are split along the season. So almost fertigation system that must be done. But in any case, I fully agree that you can use both systems, but would be easier to work with a small open base.
And so, I'm still receiving, by the way, also in my mail account, some questions. So just a last question for Davide. Does anybody know the reason why controller rootstocks are not being used widely? Can we give an explanation?
Sorry,
Can we give an explanation why controller rootstocks are not being used widely?
Okay, you know, generally speaking, you have to think the best fruit production, in which kind of shoot it is on the middle vigor. So it means fruiting shoot. It means 30, 40, 50 centimeter long. So you need to have this as a basic model. And this must be renewed in a regular way along the branch, along the limb. So this means that it will be very difficult if I have a rootstock that stops the growth very early. It will be very, very difficult to maintain this type of growth year after year. You know, for the first 2 years, it will be easy. You get a big success. But then after, it will be very difficult to maintain this kind of renewal of the fruiting shoots. For this reason, in my opinion, it's very difficult to use appropriately the dwarfing rootstock, the controller.
I very much disagree.
Okay, sorry.
I've been working with controller rootstocks now for 20 years because we developed them. It is true that the shoots may be slightly less long. We're not talking about rootstocks that dramatically reduce the vigor so you get really short shoots all the time. We have no trouble with adequate pruning and doing renewal wood pruning, like somewhat like you were recommending, that we don't have any problem maintaining good shoot extension growth, fruiting shoots with. And I had the same concern when we started looking for size controlling rootstocks. I was concerned about that, but as long as we don't go to really dwarfing rootstocks, like controller 5 is too dwarfing, but controller 6, 7, and 8, they all almost a similar type of rootstock. I mean, amount of size control, produce a very nice tree, and we've had trees producing with them for 10, 15 years now.
I agree. You know, Ted, I'm not an expert about controllers, sorry, because we don't use here. But I was thinking about the possibility to use a dwarfing type. You know, that is, in my opinion, very difficult for peach. That is my opinion, because really, in our condition, it would be very difficult to maintain the renewal. But I agree with you. You know, if the vigor is slightly a little bit more, it will be the equilibrated situation. So it's what I'm trying to suggest to people. Try to find in your environment the rootstock that has a medium vigor. Don't look just for the very dwarf type. That is my suggestion.
Yeah, no, I agree. Medium vigor, and the benefit of the medium vigor is it decreases the number of water shoots. The water shoots are the bane of peach growers' existence, and so if you can reduce the amount of water shoots, which we do with modestly controlling rootstocks, and not do a lot of heading cuts like you suggest, I think that's we can get.
Ted and David, I have to give to give the floor to Greg 1 and Greg 2 about this issue. So Greg Reichhardt first, please.
Okay, I guess the question is why aren't they being used more? We just haven't been tested enough. They look good. 6 looks very good in our trials in North America. So I think they do have a place because the fruit size is good, and what Ted mentioned, you don't get too much shoot extension. So it's just, it takes so long sometimes to get adopt a new rootstock, but I do think it has some of them have a place. So that's just my observations. I've seen them for about 15 years now.
Can I add something on Greg's comment? I think rootstock trials have been kind of artifacting the performance of vigorous rootstock. So when you are evaluating a dwarfing or semi-dwarf rootstock compared to a vigorous on an open vase, of course, you're not going to have a performance. And the other thing is irrigation scheduling is not adjusted. Usually we fail in rootstock trials because we do same irrigation scheduling as with the vigorous ones. And this is where we kill dwarfing rootstocks. In my end, this is what I'm facing here. I'm in a very dry climate, and I'm getting all the advices from all of you guys, but I think this is something that we kind of not touching it too much, but I think Controller 6, we like it too.
Okay, and can we have the viewpoint of Gregory Lang? Greg?
Well, I actually don't have anything further to add since Ioannis just made that statement. I agree wholeheartedly, and also agree with everything that Ted has said. I just wanted to say that Davide, I think those points need to be taken into consideration.
You don't agree with Greg Riegert?
Greg Rieger just said it hasn't been tested enough.
Who does?
I think we're all on the same page.
He knows what he's saying.
Okay, so I think that we are now moving to the concluding remarks for day 1. If we skipped some questions, we have still 3 days of meetings, intensive meetings. So thank you very much for your attendance. I will give the floor to Ted to make some concluding remarks.
Well, thank you very much, George. I really enjoyed this session. Maria, I learned a lot about mutations and especially about chimeras. I appreciate that. And it's no question that somatic mutations have had a huge impact on our industry. And that's, you know, that's how we get late this and late that and early this and early that. And so it's great. Ksenija, you guys are always generating more information and more of those figures and diagrams and so on. I think you're right. We're going to increase the breeding efficiency with some marker selection, but they're really tough. I do a prune breeding program. I'm overseeing that. And to make decisions when you don't know very much about the genetics of something, it's hard to throw stuff out because there's a lot of unknowns but once you have that confidence, it makes some sense. David, you covered a lot of territory. And I think we're going to always have disagreements or different philosophies about training systems, and the part that gets a little bit lost, I think, in the discussion sometimes is that each of us operates in a different socioeconomic situation, and we're all dealing with, I mean, the pressures on my growers are very much different than the pressures on your growers, and I can hardly even say our growers anymore, because in California, we have relatively few independent growers, and probably 80%, Kevin Day and I quite often comment that 80% of our production, and you see that huge production in California, is done by about 12 corporate farms. And so that's a whole different, and you have the same thing in South Carolina. You have some small growers, but then you have that one huge grower, Titan. And you have a whole different mentality with these different types of growers. And so big diversity we have to deal with. In California, it'll be interesting to see the future because in terms of breeding and the whole peach industry, because at the University of California, at the moment, there's not a single faculty person in our department working on peaches or nectarines anymore. Because we lost our funding from the industry. The industry voted the funding out. And the breeders we have, Tom Gradzel is, no, I shouldn't say, I'm talking about fresh market peaches. Tom is still working actively in the cling industry for processing. But so things are changing dramatically here. It'll be interesting to see how the breeding goes, because some of the breeding programs, we don't have a private breeding a public breeding program in either USDA or the university in breeding peaches anymore, peaches or nectarines. Other than Tom's, which is for the cling peach industry, the processing industry. And several of the other breeding programs have also been bought up by these conglomerate corporate farms. And once they realize that those things aren't paying so well directly in a short term, some of those things are already falling by the wayside. So we got some really interesting things going on. It'll be, I mean, I'm impressed. You guys come up with, there's 700 cultivars in the last, how many, 20 years or so. It used to be that California was leading a lot of that stuff, and we certainly will not be doing that anymore. So, and Ioannis, I'm really curious about some of the correlations that you come up with related to crop load. And yeah, all very interesting this morning. I was wondering how much new stuff there was going to be since the last Peach Congress in Romania, but there's plenty of peach new stuff, and I'm looking forward to next year because I think there'll even be more new stuff between this year and 1 year from now. I thought George, actually, I personally thought George might have made a mistake by doing this, because I was wondering who's going to be the speakers next year, the plenary speakers. So, but I think there'll be plenty, and it's interesting, and I really enjoyed today. Thank you very much, George, and all of you folks. And you did a fantastic job for staying on time, except for Ioannis.
I was
Ted,
playing in my field, that's why.
Ted, thank you very much. I think there is a critical mass of speakers, and one of the reasons of this webinar was to keep the momentum live for the next year's symposium, and of course to give the floor to young promising researchers like Ioannis and some others who will follow the next days. So I would like to extend my gratitude to all attendees, all speakers, Ted, to you personally, for excellent coordination of this session. And stay tuned. Tomorrow we have talks about metabolic profile and phytochemical properties about peats, nutritional content, and special reference to flavor. Also, we have the talks about sustainable orchard fertilization, and the key very hot topic about how we can reduce the production cost of peats. This is a key issue, I think, for all the peats industry. So stay tuned. See you tomorrow, 5 p.m. Central European time. Thank you very much, everybody, for the session today.
Thanks.
Take care.
Bye, thank you.
Ciao.
Thanks.
Take care.
Thanks, George.